that how useful the program is going to be, and I hope that I'll keep up the name of DNX Academy, and I have chosen this topic for the demo class because this is tumor biology or molecular biology, because this will give some versatile idea. Okay, when you approach the individual topics, when you study for a surgical onco MCH entrance examination, when you study the individual topics like CA colon, CA stomach, CA breast, or any other topic, it will give a complete idea, complete picture. When you approach the molecular basis of each tumor, I think it will make you this class, which I'm going to handle for another 1 hour or so, will help you to run through the topics of molecular biology of the individual topic. So, and it is easy to rule out many options if you have a wide knowledge of the molecular biology and the genetics involved in the tumor generation and tumor progression. Okay, so I'm Dr. Arun. I'm working in Kilpak Medical College, Chennai. And first of all, I welcome all of you to DNX Academy. It's going to be the first surgical onco class. I feel so much privileged to handle the first class, and I hope I'll make I'll give you some useful time for another 1.5 hours or so. So, how many of you are there? Shall I start the
Yes, sir, you start. Already 14 people are there. You start. It's live on Facebook.
Okay. So I'll be having some questions in between. So after each subtopic, I'll be giving some sample questions that can be answered by you, and I can see in the chat, and I can reciprocate accordingly. Okay. So going to the first slide. Okay, is tumor biology and genetics. So these all terms you all are well aware of, and neoplasia is new growth, is uncontrolled proliferation. It can be used for both benign and malignant tumor. A tumor is something to do with the inflammation. Okay, Ruber-Keller tumor. And now in oncology, this tumor is interchangeably used with neoplasm. These all you are aware of. So there is now nothing going to be asked in these kind of stuff. And cancer is absolutely, usually it indicates malignant tumor. Okay, so in cancer generation, the transformation is the important process. That means the normal cells, they acquire the malignant characteristics, that is, they invade the tissues and metastasize. And each step in this transformation reflects one or more genetic alterations. I just remember this, I'll be using this concept when we study about the individual molecular biologies of the cancer. So one or more genetic alterations can be a one genetic alteration, or it can be more successive genetic alterations of the tumor suppressor genes or oncogenes, and finally it will lead to a so which will confer a growth advantage to the normal. Yes, this is just about the. I put this slide because maybe just refreshing some old terms. By the time more people also will join, that's why I just started with. In any starting should be absolutely complete. So this is the first question. Completion of Human Genome Project has revealed that human cells have a repertoire of genes of what approximate number? So 2,500, 25,000, 250,000, and can anyone answer? Absolutely. Dr. Hari Prasad has answered perfectly. This is 25,000. Even though there are 25,000 genes approximately present in the human cell, only a few thousands they tend to express at one point of time. Not all 25,000 genes are expressing at any given point of time. Okay. So analysis has revealed 25,000 expressed genes in human cells. However, only a few thousand of these seem to be expressed at any given point of time. Yes. So coming to the epidemiology. So just 2 slides only. I'm not going to cover broadly about the epidemiology. Okay. So as you all know, in men, the common cancers include CA prostate, lung, colorectum, and urinary bladder. In women, predominantly carcinoma breast, followed by lung, colorectum, and uterus. In literature, certain literature says that lung, in absolutely there is no doubt that in women, CA breast is the most common tumor, and followed by lung. And in men, there is only a marginal difference between, it is about the country-based studies. Accordingly, the global burden is not given accurately in any of the literature. But previous literature says that maybe a decade ago, the lung was the most common cancer in men, followed by CA prostate. Nowadays, most of the literature, they commit that CA prostate seems to be the most common cancer. This is not going to be an absolute number. This I feel that these are all the not the like quality questions that can be asked to test the knowledge of a student. But still, these kind of stuff are asked occasionally. So just I put this slide to remind that. So a good examiner should not ask this kind of stuff. But in women, there absolutely carcinoma breast is the most common cancer. And in men, for time being, if at all a question is asked, you can go for CA prostate. And both in men and women, lung is the second most common cancer, and lung is the most common cause of death due to cancer in both men and women. So if it is going to be a question like which is the carcinoma which is most commonly leading to death due to a cancer, that is going to be CA lung in both men and women. If it is going to be asked combined also, it is going to be lung only. Okay. So regarding the global burden, why at all cancer is increasing or decreasing, and it is due to the following factors. Okay. So in the growth and aging of the population seems to be aging because of the good control over the medical morbidities. Nowadays, people are able to live more than 65 years, 70 years. So more the people in the age group more than 65, 70 years, absolutely, cancer is a disease of old age, and absolutely the global burden of the cancer is going to increase. And regarding the developing countries, so there is an increasing encroachment of a modifiable risk factors like cigarette smoking, Western diet, and physical inactivity, leading to your obesity and other stuff, which may lead to increase in the number of cancers. These are all the reasons why the global burden of the cancer is increasing. And relatively, there is a slower decrease in infection-related cancers. Now, though there are infection-related cancers reported, there is a very slow decrease occurring due to good vaccination, for like example for vaccination for HPV, or you have a good eradication treatment for H. pylori. Like that, infections are being controlled well. There is a slower decrease in the infection related cancer. Fine. So regarding aging and cancer, I told you that so it affects mostly 65 years or older. When there is going to be a population increase in more than 65 years or so, that's going to be having a more incidence. That's all. So obesity and cancer, absolutely, everybody is aware that obesity leads to certain types of cancers, and usually it leads to a colonic cancer, breast cancer in a postmenopausal woman, especially. We will see why in later slides. And endometrium, kidney, esophagus, gastric cardia, pancreas, gallbladder, and liver. So these are all the tumors. So most of the important solid tumors are affected by obesity. So obesity may produce a metabolic and endocrine effect, which will lead to an alteration in the peptide and steroid hormones. Most commonly, you are aware that estrogen is one of the steroid hormones, which is produced more in postmenopausal obese women. This estrogen can cause CA breast and CA ovary. Sorry, CA endometrium. So obesity and carcinoma. So this is going to be a flow chart. The same thing what I told you. So there is an increased adipose tissue, which will lead to increased circulating levels of the free fatty acids. So these tissues now they start using fats rather than glucose for energy production. So what happens to glucose? There is going to be a decreased metabolism of glucose, leading to hyperglycemia. This hyperglycemia, it leads to functional insulin resistance. That means even though there is an insulin, even though there is a glucose, the cells are not able to utilize the glucose because it is preferably taking fats because of the sufficient availability of fat. So what happens? It causes, because of the insulin resistance and hyperglycemia, though there is hyperglycemia, and insulin resistance. There is an increased secretion of insulin from the pancreas, which leads to a chronic hyperinsulinemia. Epidemiological studies and uh, certain metabolic pathways studied in the cancer pathways, they indicate that insulin and insulin-like growth factors are very much notorious. If at all they are present in a chronic hyper form, they may lead to certain type of cancers like CA colon, CA endometrium, and some other cancers like CA breast also, right? And regarding the postmenopausal women, increased adiposity, postmenopausal obese women, because of the circulating, because there is going to be an increased production in the peripheral fat tissue, there is going to be increased circulating estrogen levels, which will lead to a CA breast with poorer survival. So in case of a postmenopausal women with obesity, the chance of having a poor survival and possibility of increased recurrence is high. Understood? So that's about some idea about the introduction and the epidemiology of the cancer. And now we are going to the proper topic. I hope some more people have joined, and it is going to be the beginning of the tumor biology. Okay. So as surgeons, we are tuned to treat only the solid tumors. We don't have to do with any other tumors like hematological tumors and so on. So we treat solid tumors, we do surgery, okay, and we should be little aware of the neoadjuvant treatments and adjuvant treatments before surgery and after surgery. So that all going to be, so we should have some reasonable idea about the pathways and pathological processes, physiological processes which are involved in the tumor initiation, tumor progression, and spread of tumor from the primary site to a secondary site. So solid tumors, it usually consists of neoplastic cells and stroma. It is not going to have only the cells. It is going to have all any cell, even if tumor cell requires a scaffold to hang on. So neoplastic cells and stroma both constitute the solid tumors. So both are interdependent. That means interdependent means so myself and you are interdependent, because I am teaching you and you are learning from me. So you are going to communicate to make a good understanding of what to happen in the further time. Okay, so that's going to be interdependent. That means neoplastic cells and stroma, they speak to each other. They send the messengers. They send the growth factors. They send the growth inhibitors, and they decide what to do and what are all the possible ways, good ways to increase the tumor growth. And if at all you are going to treat the tumor, how we will plan together and give a resistance, and we will just neglect the treatment. We'll just throw away the treatment given by the either in the form of chemotherapy or radiotherapy, immunotherapy, whatever it is. And they speak with each other, and we will plan and disguise ourselves in what way it is safe and safe to enter the circulation in the blood circulation or lymphatic circulation, so that we can go and feed somewhere else in the body and cause a tumor generation in that part also. So neoplastic cells and stroma, they both are interdependent, and they speak with each other, they communicate with each other by means of growth factors, by means of inhibitors, and they decide what to do to further plan for a growth of tumor. Okay, tumor cells, they are very intelligent, like they talk with all these stuff, and they have good communication capabilities, so that they will try to catch hold of a good environment which is suitable for their growth. Fine. This stroma is not going to be a single thing. Okay, stroma means it's not a single thing. Stroma has so many cells, you have so many proteins, you have so many extracellular matrices, which has proteinases, proteases, integrins, pericytes, endothelial cells, and so many stuffs are there. And each individual cells and each individual chemicals, each individual proteins, they will do their part. Some of them send inhibitory signals to the tumor cells. Some of them send growth factors to the tumor cells. Accordingly, it is going to be an interdependent meeting. Finally, a outcome will come, whether to proceed or it's going to destroy the tumor. So what happens? See, in a tumor, it's not going to be a single step process. It's always a multi-step process. You have successive genetic changes. So genetic changes are the initiating points which leads to So that successive genetic changes occurs in one group of cells, maybe a single cell also. So which provides a physiological growth advantage when compared with other normal cells. What is this physiological growth advantage? We will see in the further slide. Okay. What does this do? This physiological growth advantage, they make the normal cells get converted to the cancer cells. So this is regarding the cancer cells. Okay. What about the nearby stroma? So the stroma also undergoes some physiological changes. Okay. So which will perpetuate or which will increase the tumor progression. So I told it is going to be a stepwise progression. For example, in melanoma, so step one is going to be a common melanocytic nevus that may be due to a small one particular genetic change, followed by a dysplastic nevus, and followed by a radial growth phase of melanoma. What do you mean by radial growth phase? For example, it's going to be the skin. So this is going to be the melanoma. Okay, this is going to be the melanoma. What happens is that stepwise. First, there is going to be a common melanocytic nevus, followed by dysplastic nevus. There is evidence of dysplasia, followed by radial growth. Radial growth means it is going to extend along horizontally, radially. Okay, it is not going to come deep or it is not going to project outside. So this is going to be a radial growth phase, followed by now that vertical growth phase. This is going to be the melanoma. What happens is that it's going to grow down and it's going to project outside also. Okay, the tumor size will increase. Accordingly, correspondingly, there is going to be a deeper extension also, which will invade the blood vessels and which will also invade the lymph vessels. That will lead to a lymph node metastasis and a bloodstream metastasis. This is how it goes. You understand? So followed by the metastatic melanoma due to the invasion of the blood vessels and lymphatic vessels. I hope you understand. It is going to be a stepwise progression. No cancer cell is going to, so when the cancer cell occurs, it is not going to immediately metastasize or it is going to spread to a different organ, right? It is going to be a stepwise progression. So the outcome depends on where we are going to act. If we are going to act in the radial growth phase of melanoma, it's well and good because there a possibility of metastasis is very less. When the melanoma reaches a vertical growth phase, the chance of having uh, reached the blood vessels and lymph vessels is more, so possibility of metastasis should be always kept in mind right before the treatment. You have to give a systemic treatment and then followed by a local treatment. Okay, coming to tumorigenesis or the generation of tumor. Okay, so how do that occur? It is not going to be a single physiological process which is going to cause a tumor generation. It is going to be variety of physiological processes, or there is going to be distinct capabilities of tumor cells to progress into a tumor as such. Okay, so what are all the distinct capabilities a cell should have to become a cancer, right? To undergo a carcinogenesis, I mean. So the first one is going to be
Arun sir. Arun sir, there is some issue in voice. Arun sir. हेलो हाँ मैं उनको बोल रहा हूँ उनका आवाज नहीं आ रही है नहीं उनको पता Arun sir, are you able to hear us? Arun sir. Arun sir. Arun sir. Arun sir, are you able to hear us? Arun sir, are you able to hear us? Your voice is not coming, sir. Arun sir. Okay, because no, you are not listening. That's why I removed. Okay, sir, I will do something from here. You use your desktop, sir. Otherwise, laptop, sir. Yes, sir.
I was talking for 5 minutes without knowledge.
Yeah, no
I'm
issues, sir.
sorry about, dear students, sorry about the obstacle. And I'm back, and we are going to start again with the tumor genesis. Okay. So tumor genesis, that means tumor cell has a distinct capability. So it has different capabilities of physiological process it is capable of, and it is able to initiate the tumor. It is going to progress through the different stages, and it is going to invade and metastasize. So these are all the different stages. First is sustaining proliferative signaling. That means I told you there is going to be signals from the, it is going to be a communication between the tumor cells and the stroma and immune cells, etc., by means of growth factors, inhibitory factors, and other types of factors, other stuff, and which may be coordinated by the tumor cell, and which will favor the growth, which for which the tumor cells will give signals in such a way that it will favor the growth of the tumor cell. How it is going to favor it, we will see in the forthcoming slides. And it evades the growth suppressor. So the growth suppressors here is in any cell cycle, there is going to be checkpoints everywhere, or 2 or 3 checkpoints to see whether the cell which is undergoing the cell cycle is the normal cell, DNA stuff is fine, or all other stuff is fine, and then only it allows the cells to progress through the cell cycle. There are different checkpoints mediated by different growth suppressors, which are mediated mainly by the tumor suppressor genes like P53 or retinoblastoma gene or neurofibromatosis gene, etc. These are all the tumor suppressor genes which will have a checkpoint, which will produce a checkpoint to suppress the growth of the abnormal cells. But these tumor cells, they will do something. They disguise themselves. They will show themselves as a different thing, and they try to evade all these growth suppression. And they avoid immune-mediated destruction by certain mechanisms. I'll tell you how in the forthcoming slide. They enable a replicative immortality. That means replicative immortality means immortality means it is not going to die. Replicative means it is going to divide, but it's not going to die. That means how do they enable this mechanism? By means of some substance which will usually after certain divisions the cells will normal cells, differentiated cells, they will reach senescence. And that what do you mean by senescence? That when they reach senescence, there are not going to be any further division. The cells will either die or they will be remaining like that for some time. Once there is going to be any abnormality, they will apoptosis or something. But here, due to some mechanisms, these cells will have a replicative immortality. They'll be going on dividing and they are not going to die also. And there is going to be a tumor promoting inflammation. Even though the immune cells are going to destroy any abnormal cells, they overcome that, and these tumor cells will send signals as such that these inflammatory cells or the immune cells, they will produce a lot of cytokine like stuff, chemokines, cytokines, and they promote inflammation in the site of the tumor generation. This inflammation itself will promote the growth of tumors, and let us see how in the future slides. So here about the how a tumor generation occurs, tumor initiation and the progression. Now that how it is going to invade the blood vessel, how it is going to invade the lymphatic vessel, how it is going to invade and go to a different place and get settled there to produce a tumor there. How do they metastasize? So they activate some mechanisms to undergo all these processes, and they try to produce blood vessels for their own growth. Nutrients are required, whether for normal cells or for tumor cells, nutrients are 100% necessary. And tumor cannot grow without blood supply more than 1 millimeter. So it can grow up to 1 millimeter by harvesting something else. But if it grows, it's going to grow more than 1 millimeter. It absolutely requires blood vessels for the nutrients to get in. So how do they induce that? By means of certain vascular growth factors. That we'll see what all the factors are going to cause that. And genome instability, and they try to resist the cell death like apoptosis by undergoing certain mutations. And the energy processes inside the cell can be altered in such a way that the cells will just proceed with the proliferation and invasion and the spread to elsewhere. So we will see the individual characteristics one by one. And first is sustaining proliferative signal. So I'll go to this slide. So I told tumor is there and nearby stroma is there, which are both are uh, going to be together to form the tumor, total tumor. This is a tumor cell, okay, and this is going to be stroma. So yeah, complete organization of multiple tumor cells and stroma, they form a solid component of the solid tumor. So what happens is that these tumor cells, they secrete certain substances. They send some growth factor. They send some messengers, chemical messengers, and they have their own. They send a messenger, say alpha, and they have a receptor for alpha, and they promote themselves for the further growth. So this is regarding the autocrine function. The tumor cells itself will secrete something, will send signals, and which will influence on the further tumor growth. And this is called autocrine. So tumor cells itself increases the growth of tumor cells, right? So what do you mean by paracrine? Paracrine means the tumor cells send signals. So here the arrows are in both sides. This tumor cell sends signals. They want something, something, and they have a good coordination with stroma. And they sometimes disguise themselves. They don't tell the like this what this no. They see there is going to be an abnormal cell. There are going to be infiltrates from the immune cells. Okay, immune system comes to play. And try to throw away what is abnormal. And but what happens is that it sends signals as if these signals will just cheat these immune cells. They just disguise themselves that they are not tumor cells, and they'll give a different information. And these cells will also produce. These cells also will produce some growth factors, which will be further increasing the chance of tumor generation. Likewise, in stroma, you have different stuff like mast cells, parenchymal cells, and endothelial cells. And most important is extracellular matrix, which has proteins like different proteases and different proteins like integrin, and you have pericyte, and all these stuff. They send different inhibitory signals as well as growth signals and stimulatory signals, and all these stuffs will counterbalance each other and finally decide whether the tumor is going to grow or not. So these inhibitory signals will be overcome by the stimulatory signals at one particular point of time. At that point of time, the tumor is going to further progress. Okay. So tumors may produce growth factors for which they have the receptors in an autocrine fashion, and it can be paracrine factors. They stimulate the surrounding normal tissues, and these normal tissues provide growth factors for the tumor. And tumor may become hypersensitive to growth factors by upregulation of growth receptor. That means even though there is now that there is not going to be any stimulation of the growth factors in the receptors, these receptors they will alter in structure, and without being good external stimulus, they try they just activate the further downstream pathways to go ahead and lead to a proliferative signal. So these are all the cells, and these are all the factors secreted in stroma. Different types of cells are there, and the factors are growth factors, growth inhibitors, nutritional factors, angiogenesis factors on cytokines, and degradative enzymes. All are secreted, and so these secretions are all communicated with this stroma communicates with the tumor cells, and finally they decide what to be secreted and whether the tumor is going to grow or not is decided by. So this tumor will overcome all these inhibitory signals by sending different messages and by sending different signals, and ultimately leading to a tumor development. So same thing occurs in the infiltrating cells like cytokines, chemokines, cytolytic factors, angiogenesis factors. Everything will be secreted by the infiltrating immune cells also. So though I told the different types of signals are produced by tumor cells for the further growth of the tumors. Initially, during the initial stages of the tumor progression, it is going to be mainly paracrine factors which are going to cause the tumor progression. And at a later stage, intermediate stage, you have mainly autocrine fractures, and at last, the tumor becomes mainly acrine. What do you mean by acrine? So these tumor, they become independent of any growth mechanism. They do whatever they want. They reach a stage that nobody can stop them do anything unless we surgeons or doctors can do something. They reach a stage that naturally occurring protective mechanisms, they cannot stop their growth because they derive everything independently. So initially paracrine, after that autocrine, and finally it becomes mainly acrine. Okay. The same questions can be framed like this. Which of the following growth mechanisms are dominant during early stages? Which of the mechanisms dominant during the late stages? Accordingly, you can give the answer. Answer here is paracrine. So that is regarding three processes I told. Okay. Paracrine and upregulation of growth receptors now. So they may become independent of the growth factors. They need not require a growth factor at all because of the somatic mutations activating the downstream pathways. I will tell you, show you the pathway and I will make you understand how it works. Okay, I'm saying, so this is the slide. Okay, so for any growth to occur, any proliferation of cells to occur, there requires something called the growth factor. There is going to be a receptor in the membrane. Following that, the receptor activates the growth factor activates the receptor, and further it activates different pathways which leads to a cell proliferative signal and angiogenesis, protein synthesis, etc. So what happens here is that, so when the growth factor is going to be excessively stimulating, what happens is that this process will be excessively activated and there is going to be further activation of the downstream pathways and leading to cell growth and protein synthesis. But sometimes these growth factors and the receptors may be absolutely normal. Problem with the growth factors and sometimes problem with the abnormal receptors, alterations in the receptors. Even without growth factors, these receptors will stimulate the pathways and leading to increased cell generation. And so even though here the third mechanism I told you, all these stuffs are normal. What happens is that in the downstream pathways, you have mutation in any of these protein, any of these genes, and they start activating the further downstream pathway and leading to proliferation and survival. And this is how it works. There are two main pathways you should be aware of in this kind of growth factor mediated, receptor mediated pathway. Commonly, most of the cells in the body, they undergo by one of these two pathways. There are some more pathways which are present. I don't think it is required for our it is required in our scenario to appear for the surgical oncology examination. I just gave two examples. One is the MAPK pathway, MAP kinase pathway. Another one is the PI3K AKT mTOR pathway. Okay. What happens here? So this is the MAPK pathway. So the receptor tyrosine kinase is over here. It activates the RAS genes, RAS genes, and which following that RAF, BRAF, CRAF, these RAF genes are activated and proteins are generated and followed by activation of MEK and followed by activation of ERK. This ERK enters the nuclear membrane and go to the chromosomes and go to the DNA material, and it just tries to influence the generation of oncogenes to start the cell cycle, to enhance the cell cycle by producing cyclin D. These cyclins and cyclin dependent kinases, I will explain you what it is when we come to the cell cycle. I can hear some noise. Can you please mute your audio? Fine. Okay. So this is the MAPK pathway. What happens here that even though there is not going to be much stimulation here, sometimes what happens here, this BRAF or MEK, these genes, they undergo mutation. Okay, they undergo mutation and they just activate the further downstream processes leading to cell proliferation. This is the same MAPK pathway which you have seen now, and this is the PI3K and AKT mTOR pathway. Here also the receptor tyrosine kinases like HER2 or EGFR, they just activate the PI3K and which has further pathways downstream, PIP2, PIP3 and followed by AKT. Okay, this AKT just undergoes certain process. TSC1 is the tuberous sclerosis 1 and 2. These genes are activated, further proteins are generated, and ultimately mTOR is activated. Okay, mammalian surrogate target of rapamycin is activated, and you have influence on cell growth, protein synthesis, and angiogenesis. Ultimately, it will result in the abnormal proliferation of the cell. These are all the normal pathways which are present. These normal pathways will be enhanced, will be activated in the tumorigenesis. So that is how it happens. Either like growth factor is more excessively stimulating the receptor, or the receptor itself alters and produces excessive signals to the downstream pathways, or there may be mutations in any of the downstream genes or downstream proteins which will further activate the pathway which are going to be distal to that, and which will be ultimately leading to a uncontrolled proliferation. Okay, so there are checkpoints in between. For example, in the PIP3, there is a tumor suppressor gene called P10, okay, which will suppress the pathway. Once there is going to be an activation here also, if these genes are going to be fine, it will try to resist the generation of further cell proliferation. But sometimes in Cowden syndrome and all, the gene like P10 is mutated. So what happens is that the control over the intermediate, the downstream pathway is not there. So what happens is cells undergoes uncontrolled division. So there can be a growth factor, there can be a receptor alteration, or can be a mutation in any of the downstream genes or downstream proteins. It can be an oncogene or it can be a tumor suppressor gene. Fine. I hope you understood all this. If someone puts thumbs up, I'll be happy. Open the chat. Yes. This is how it goes. So what we can do is, when we analyze where the mutation is going to occur, it is going to occur in the growth due to the growth factors are going to be occurring in due to the receptor. You can provide some drug here to block the receptor, or you can, if there is going to be mutation for like specific mutation in the BRAF like V600E in case of melanoma and lung carcinoma, you can use BRAF inhibitors like pembrolizumab, dabrafenib, etc. It's going to be a mutation in MEK. You can use MEK inhibitors like trametinib, binitinib, etc. And in ERK inhibitors are also available. So this is how it works. This is how we use targeted therapy in a cancer. So we go to the molecular pathway where exactly the defect is noted. We just inhibit but at that particular point. So before proceeding further, let us have some revision about the cell cycle. Cell cycle, all of you know, you have gap 0 phase, gap 1 phase, and synthesis phase, gap 2 phase, and mitosis phase. Gap 0 phase is the resting phase. The cell is not going to do anything, just rest on. There is a requirement, it will come back to the cell cycle to the G1 phase. G1 phase is also called as interphase. In there is going to be an early G1 phase and late G1 phase. In late G1 is the checkpoint which is going to occur. In early G1, there are going to be some other processes that are going to happen. I'll tell you. In late G1, there is going to be a burst of RNA synthesis and many enzymes which are required for the synthesis phase. In S phase, synthesis means it is DNA synthesis. They are all manufactured. They prime themselves to undergo a DNA synthesis. Following an S phase, you have a gap 2 phase. Here the DNA synthesis comes down, but still the RNA synthesis and protein synthesis are proceeding. Certain microtubular precursors of mitotic spindle are produced because they prime the cells to undergo mitosis. They just start the process what is required for the mitosis, followed by a mitosis phase. We will see what are the phases in mitosis later. So just listen to this slide carefully. Okay, lot of questions can be asked in this slide. Though this cell cycle is known to all of you, you should have some idea about the factors which influence the cell cycle. As such, cell cycle itself, they are not going to ask any questions, but the factors which are going to influence the cell cycle, which are going to inhibit, which are going to stimulate, and what way it is restricted, everything can be asked in from the cell cycle. Okay, so you have, cells undergo mitosis and cytokinesis, and they come to a G1 phase. You have early G1 and late G1. Okay, so in a cell cycle, you should have an idea about at least 3 or 4 substances like One is cyclins. These cyclins are mandatory. These are all stimulatory. They are all stimulatory for the cells to undergo division. So they stimulate, they just make the cell enter the cell cycle, undergo division. This cyclin is cyclin D itself is an oncogene actually. Proto-oncogene, if it is going to get mutated, it is going to result in an uncontrolled proliferation. This cyclin, they cannot act independently. They required enzyme like an enzyme called cyclin dependent kinase. They are all serine threonine kinases. They are all protein kinases only. Okay, this cyclin always requires CDK, that is cyclin dependent kinase, for to act upon in the cell cycle. So this CDK cyclins and CDK combination is specific. For example, cyclin D combines it only with cyclin CDK4 or CDK6. Only after joining with CDK only it is going to stimulate the cell cycle. Cyclin as such individually it doesn't have any capability to stimulate the cell cycle. So in G1 phase, the cyclin D and CDK4 and 6 they combine together. What do they do is that normally this retinoblastoma gene, that is RB gene, is the tumor suppressor gene. Usually it is in a hypophosphorylated stage. That means less phosphorylation. During the hypophosphorylated stage, it has a growth factor called E2F. It is attached to a growth factor called E2F. So what does this cyclin D and CDK4 combination? They come and phosphorylate the retinoblastoma gene. They phosphorylate the retinoblastoma gene, and just the retinoblastoma gene gets separated from the E2F, and E2F is going to be the stimulating factor in the late G1, and it is going to send the cell for the further division. When the retinoblastoma gene is mutated, or retinoblastoma is deactivated by phosphorylation by the cyclin D, it enters the synthesis phase. So from G1 to S transition, the cyclin E and CDK2 combination is very important for the transition from G1 to S phase, S synthesis phase. Throughout the synthesis phase, the cyclin which is active is cyclin A. So G1 S, this phase, CDK2 is the cyclin dependent kinase involved, and cyclin A and cyclin E are involved in S phase. So once it enters the G2 phase, the same cyclin A combines with CDK1 and makes the cell proceed with the G2 phase. And following G2, the cells enter mitosis. They enter division. They are going to become divided into two daughter cells. In that place, this mitosis is completely, they are controlled by CDK1 and cyclin B. Just remember two important things. Cyclin D, CDK4/6 is in G1 phase, and cyclin E and A are in S phase. Cyclin B, remember cyclin B is in mitotic phase. That will do for your question's sake. So just have some idea. These cyclin and cyclin dependent kinases, they are mandatory for the cells to stimulate the cell cycle and make the cells undergo division. So these cyclins at different steps of the different stages of the cell cycle, different cyclin and the CDK combinations are prevalent, and you should know where it is prevalent, and questions can be asked in that. Okay. So how this cyclin D is stimulated? Even in the previous slides, you remember? See, all these pathways are activated, and they go and enter the nucleus and just stimulate a cyclin D1 production. This cyclin D, what it will do is that it will just push the cell into the G1 phase and lead to an uncontrolled division. Understood? This mitosis, okay, it is not going to be a single process. This mitosis, as you all know, just I'll refresh the stages of mitosis, because in Devita review by Ramasamy Govindan, the question has been asked. Tell me the correct order of what is the correct order of mitosis, and they have given 4 choices. That's why I put up this slide. After G2, there's going to be a prophase. What happens in prophase? The spindle fibers appear and the chromosomes condense. In pro-metaphase, the spindle fibers attach to the chromosomes. In metaphase, the chromosomes they align, and in anaphase, they divide the centromeres and sister chromatids. They move to the opposite. In telophase, nuclear membrane is reformed and chromatins distant. And what happens after telophase is the cytokinesis, the cytoplasm divides. Whatever division occurring in prophase, prometaphase, metaphase, anaphase, and telophase is the nuclear division. And at last, once the nuclear division is complete, the parent cell divides completely by dividing the cytoplasm into two daughter cells with exactly identical genetic information. The correct order of mitosis is, this is, I have not framed this question. This is a question from the data review. Anyone can answer? Exactly. Okay. So which of the following proteins has an inhibitory activity in the cell cycle? I taught you about all other stuff, but I have not told you anything about this. I will tell you what it is later. Just answer this question. Any more options? Answer. Which of the protein has inhibitor? I told you can rule out, even though you don't have knowledge about P16 in 4A, you can rule out cyclin E2F and CDK4. They are all involved in the process of cell cycle. So stimulatory process of cell cycle. So the odd one out is P16, INK4A. How it is going to inhibit? This is nothing but a CDK inhibitor, cyclin CDK inhibitor. I'll tell you how it works later. Okay. So what is cyclin dependent kinase? They are ubiquitous in the cell cycle. They just undergo phosphorylation and they associate with cyclins for activity. CDK4 and cyclin D1, they phosphorylate RB protein, depression of transcription factors. And there are series of inhibitors, that is INK4 family, INK4 family. They act to decrease the activity of cyclin. They are all cyclin inhibitors, okay, of which P16 is a member. Predominantly, P16 is a member. The DNA damage checkpoints are located in which phase of the cell cycle? Anyone can answer? Please put forth your answers so that I'll straightforward only. You can try, no? I'm not asking you. Any more answers? So one answer has come, that is A, absolutely. Because SG2M, they are all not checkpoints. The checkpoint, three major DNA checkpoints are G1S, G2M, and S phase. So these pathways promote cell cycle arrest. Okay, G1S, the main factors involved is the P53, and I told you cyclin inhibitors like P16INK, etc., P21 will be influenced by P53. All these stuff, they will try to trace out the abnormal cells and try to induce apoptosis and throw away the abnormal cells. In G2M also, there are certain processes which check. Here also P53 plays a role, and S phase also. Only if the DNA in the S phase, it is checked whether the DNA synthesized is normal, or if there is any abnormality. Before entering the G2 phase, the DNA will be repaired, and then only it will be sent to the G2 phase. Cyclin responsible for facilitating the progress of the cell into and through mitosis. These are all the questions. I have not framed all these questions. One or two questions I framed, but these questions all are taken absolutely ditto from the data review. Okay, and these questions were asked when I was undergoing the entrance examination in the recent past, in JIPMAR, AIMS, and in one of the NEET exams. And I think in 2017, I believe, these questions, one of these questions appeared. Anyone has answered? Responsible for progress of cell entry into through the mitosis is? Yes, just remember this diagram, no? So in the mitosis, I told cyclin B. In G1 phase, cyclin D, and in S phase, cyclin E and cyclin A. This cyclin A still continues in G2 phase. Just remember, this is DEAB, DEAB. This is DNX Academy. You can remember DEA and then followed by B. is cyclin B. This question was asked in one of the JIPMER examination, I remember. So that is regarding the cell cycle. And the second process is evading growth suppressors. Normal cells, they use multiple mechanisms to regulate negatively the cell proliferation. But these processes, how do they occur? They occur by products of activation of these tumor suppressor genes. Loss of functions of these tumor suppressor genes, they allow the cells to evade the inhibitory mechanism. I told you already in the picture. So I just took retinoblastoma gene as an example. This was the first, just remember, there can be a question, which is the first tumor suppressor gene to be cloned? That is a retinoblastoma gene. The control of cell cycle and switch from the resting state to cell division is controlled by retinoblastoma gene. How it occurs? I told you already. Okay, the cyclin D. Okay, the cyclin D comes and phosphorylates the retinoblastoma protein, and the combination with E2F, the E2F gets released from the retinoblastoma gene. This E2F joins the E2F site, and it induces it causes the basal machinery and induces the particular gene to progress for the S phase, DNA synthesis phase. This is how it occurs. Okay. This is regarding the, so when there is going to be a increased cyclin D activation, that is oncogene activation, or going to be a phosphorylation mediated by some other processes of retinoblastoma gene, it leads to, when the retinoblastoma gene is not active, it leads to uncontrolled cell cycle progression. And one more important tumor suppressor gene I want to comment here is the TGF beta, transforming growth factor beta. It prevents the phosphorylation of the retinoblastoma gene and thereby blocks advance through the G1. So what does this cyclin D do? This cyclin D phosphorylates the retinoblastoma protein, but this TGF, so I should put here TGF beta. But this TGF beta, by activating the SMAD4 protein in the cytoplasm, okay, this SMAD4 protein, they activate P16, P15, P27, and P21. These are all, this P16 we already know about it. P16, I told you, how it inhibits. It inhibits a cyclin inhibitor. This P16 inhibits CDK4 and cyclin D combination. P27 and P21, they inhibit cyclin E. And by inhibiting the cyclin D, this process is inhibited. So retinoblastoma gene is still hypophosphorylated, and it is always in combination with E2F, and it is not going to, there is going to be an inhibition of the gene. It is not going to further proceed for the division. Activation of anti-mitogenic signaling involves following molecules. Molecule or molecules? Anyone can answer? Oh, excellent. That is D. So I told you TGF beta. So TGF beta, by certain processes, it inhibits the cyclin D. So the cell process is inhibited. And interferon alpha is a cytokine produced from the cells, which also inhibits the mitosis. And upregulation of CDK inhibitors. CDK inhibitors is nothing but P16, P15, P27, and P21. When they are upregulated, it is going to inhibit the cyclins and cyclins. So all these processes are required in activation of anti-mitogenesis. Next is resisting cell death. So most of the cells, tumor cells, they are characterized by defects in the cell death signaling. So there should be a signal to acknowledge that the tumor cell is abnormal, and it will make the cells to proceed for the apoptosis. But there is a defect in the cell death signaling pathways, and they are resistant to cell death. So before that, we should understand what is apoptosis. It is a physiological cell suicide program. All of you are aware of what is apoptosis. I'll just highlight the important points in apoptosis. It can be induced by an extrinsic or intrinsic pathway. Okay, extrinsic pathway is activated by an external stimulus, okay, which will go and activate the caspase 8, and this caspase 8 activates the caspase 3 and leads to a caspase cascade leading to apoptosis. And intrinsic pathway, what happens is that, so any DNA damaging agent, for example, a chemotherapy or radiotherapy is given, it damages the DNA. So what is the gene which is essential to check whether the DNA is normal or not before further in the cell cycle? P53 is the important gene which acknowledges a DNA damage or any abnormality, and it sends signals to the pro-apoptic genes. Pro-apoptic genes like PUMA, NOXA, and BAC1, BAX, and BID. BID, BAX, BAC1, PUMA, NOXA, these are all pro-apoptic genes. There are So when there are going to be pro-apoptic genes, there are going to be anti-apoptic genes also. So the main anti-apoptic gene here is the BCL2 family of genes. So how to remember this, which is an anti-apoptic gene or a pro-apoptic gene? I just give a clue. Most of the pro-apoptic genes, they will have a vowel, A, E, I, O, U, one of the vowels in it. But in an anti-apoptic gene, you will not have a vowel. It's just a like simple clue. It need not be applicable all the time. There are going to be a question asked. There is going to be a question asked in which all the following are pro-apoptic genes except. In that case, if you are not able to remember what it is, just remember that gene which is having a vowel, A, E, I, O, U, they are usually pro-apoptic genes, and the ones which usually they don't have a vowel, that is BCL2 family of genes, they are all anti-apoptic genes. When the P53 is activated, these P53, they just stimulate the pro-apoptic genes and they just inhibit the anti-apoptic genes, and leading to a production of cytochrome C. This cytochrome C, what it does is that it joins with procaspase 9 and APAF1. This is called as an apoptosome, and which produces an activated caspase 9, which in turn produces caspase 3, which leads to a caspase cascade and apoptosis. Just remember, these are all the pro-apoptic genes, anti-apoptic genes. The pro-apoptic pathway is mediated by the P53 gene, and there is an extrinsic pathway with that the signal comes directly, and there is a conversion of procaspase 8 to 8, caspase 8, and following that caspase cascade occurs. What are the features of apoptosis? There is a question asked in the Devita review. That is why I have put up this slide. Otherwise, it's a very simple fact everyone is aware of, but sometimes you may miss the answer. So this is a normal cell. In apoptosis, once apoptosis is started, the cells start to shrink, and there is a chromatin condensation is going to occur. Yes, and following that, DNA and nuclear fragments. This the DNA and nuclear they do fragmentation. This nucleus gets fragmented, and the membranes are going to bleb off here and there. And finally, the blebbed off membranes, they go out as a apoptic bodies. So absolutely, there is no inflammation. Just remember that. That is more important. Okay. So that is regarding the apoptotic cell death. And now coming to the non-apoptotic cell death. Apoptotic cell death, just remember, apoptosis is one type. This is the type of cell death which will prevent the tumor formation, which will send the abnormal cells, which will just make the abnormal cells die, and they will just put a halt to the tumor generation. But rest of the tumors, the rest of the cell death, necrosis, autophagy, or mitotic catastrophe, they most of the times they favor the tumor formation. Necrosis can occur in infection, inflammation, and ischemia. So when there is going to be a site which is going to have a necrosis due to an infection, for example, a necrosis due to we'll just take HPV as an example, human papillomaviruses, which when they act on the cervical area or it's going to be a perineum, they are going to cause necrosis of that particular patient, inflammation and necrosis. What happens is that this necrosis, just when there is going to be an inflammation or infection, it is going to harbor the immune cells. Okay, these immune cells, they produce certain cytokines and throw inflammatory signals. These inflammatory cells are usually of myeloid origin, which will induce the angiogenesis, immune suppression, tumor invasiveness, and metastasis formation. So necrosis. So most of the times when you operate a large tumor, okay, we all are surgeons, we know that we operated many tumors. Oncology is part of general surgery. You operate large tumors. When you send it for a histopathological report, they say that there are intermediate necrosis present. So when you take a biopsy, true cut biopsy, only necrosed tissue is present, nothing else is present. That means this necrosis is a part and parcel of tumor generation and tumor progression. Just remember that. So autophagy. So there is this apoptosis is called as a type 1 cell death, and in autophagy it is going to be a type 2 programmed cell death. Okay, this autophagy is triggered by, see, when there is going to be a growth factor withdrawal, hypoxia, or DNA damage and differentiation or development that triggers this autophagy is going. What happens in autophagy is that it degrades the intracellular organelles and giving rise to metabolites that allow survival of tumor cells. In what happens is that when the intracellular organelles are like degraded, and the certain metabolites which are produced which enhance the survival of the tumor cells, even though there is going to be a stressed nutrient limited environment, because formation of angiogenesis occurs at a later stage only. Okay, immediately it won't occur. So when there is going to be a stressed nutrient limited environment, this autophagy helps to allow the tumor cells to survive. Okay, this process is usually regulated by this PI3 AKT mTOR pathway, which I have seen already. I have just explained about what happens in the PI3 kinase AKT pathway, and also by a protein called Becklin. Okay, it is mediated by a protein called Becklin. So what is a mitotic catastrophe? So this is going to be an aberrant mitosis. So going to be an abnormal mitosis caused by failure of the G2 checkpoint. I told G2M is one checkpoint and G1S is one checkpoint. And G2M checkpoint, once it is going to get failed off to block the mitosis, and the damaged DNA, okay, what happens? The damaged DNA which is going to enter can lead to a cell death. Okay. The signaling pathways involved in non-apoptotic cells are not well defined, like how it is defined in apoptosis. But there is definite evidence that all other non-apoptotic cell deaths, they favor tumor formation and tumor progression. Okay, but apoptosis is the only restriction by doing a cell death, they just stop the tumor progression. So example, so MDM2 gene, it negatively regulates P53. Okay, so just remember that even a P53, guardian of the genome we call it, even a P53 is controlled by madam. Okay, a madam controls everywhere. Madam controls the husband, madam controls the students, and even a P53 is controlled by madam, MDM. So just remember which of the following gene negatively regulates P53, and 4 options are given. Just write it as, just put your answer as MDM2 or madam2. Okay, one madam itself is difficult. There are 2 madams, so it is very difficult for the P53 to do its function. So sometimes deletion of autophagy regulating gene, I told you about Becklin, no, in high percentages of ovarian and breast carcinoma are seen. So when it is studied, the Becklin is present. So that is how we come to a conclusion that autophagy has a role in tumor progression. Type 1 cell death is also known as type 1 programmed cell death. I mean, it's also known as excellent. It is apoptosis. Type 2 cell death is also known as same 4 options. Can you answer? Type 2 cell death. Type 2 programmed cell death. Excellent, excellent. That is autophagy. Okay. Apoptosis and autophagy are all programmed cell death. Necrosis is not. Okay. Apoptosis. Okay. Type 2 programmed cell death autophagy is regulated by. Excellent. In the previous slide, I have put up it is mTOR PH3 kinase pathway. These back are all pro-apoptotic genes. BH3 also is involved in apoptosis. APC is involved in mitosis. That is a different. Anaphase promoting complex. That is in mitosis. Autophagy has a role in the following except. So what is the normal role of autophagy? Usual role of autophagy. Anyone can answer? So it is cell lysis caused by surrounding inflammation. Inflammation will be present only in necrosis. Autophagy has a role in recycling of the normal cellular component, and it has protection against aging also, and damaged protein and organelle removal also is possible by autophagy. There is absolutely no inflammation in autophagy and apoptosis. Going to be necrosis, which is going to have enormous inflammation. So coming to the next physiological property for the tumor to grow and invade, next is the replicative immortality. What do you mean by replicative immortality? I told you, immortality means it is not going to die. And replicative immortality means the cell is not going to die and it is going to continuously replicate. That means it is going to continuously divide. So there is going to be an uncontrolled division. So except stem cells and certain germline cells, normal cells, they have only limited replicative potential. So they have to undergo either senescence or apoptosis. Senescence means it's aged. Okay. Just remember that after one particular age, we may not be able to do the same, in humans. I mean, in humans, after we attain age of 70, we may not be able to do the same work what we have done in 30 or 40. So like that, the normal cells, they just divide. After some time, they either enter senescence, either we enter 80s and 90s and we'll be in a old age, or we will die. So the same way, individual cells also, either it enters the senescence or it dies by apoptosis, because after certain divisions, there may be a abnormal nuclear material present that will be detected by P53, and it activates the pro-apoptotic genes and it inhibits the anti-apoptotic genes, and it leads to apoptosis. Senescence. What is senescence? What is quiescence and what is senescence? Quiescence. Quiescence. What is senescence? In a cancer, senescence means the cells remain dormant, they will not divide, and they'll be like that only forever. But very rarely it can be reversed. But quiescence means the cell is silent, that's all. That they are in G0 phase. But any good circumstances arise, they enter a G1 phase and they come back to a normal cell division. Okay, that is the difference between quiescence and senescence. A question may be asked. There is a question interrelating quiescence and senescence in data review. Okay, so the number of doublings means number of divisions. Once the cell divides, it is going to be 2 cells. That is doubling. So this is controlled by what is called as telomeres. What are these telomeres? Usually there are 6 short 6 base pair sequences, present in the ends of chromosomes. They have several thousand repeats of the same element. Okay, what are these telomeres? They prevent the end-to-end chromosomal fusion. However, each DNA replication is associated with loss of 50 to 100 base pairs. So once the DNA, every time the division occurs, there is going to be loss of 50 to 100 base pairs of the telomeric DNA. That will reach a threshold point where, that is when they reach a critical length, the unprotected chromosomal ends they participate in chromosomal fusion, yielding a karyotype disarray that almost inevitably results in the death. So just simple. We don't need to go through all these things. Very simply, see, these telomeres are repeated. Okay, these telomeres are short 6 pair sequences. They repeat themselves. They are present in the end of a chromosome. After every division, the DNA of the telomeres gets shortened. Once the critical length is reached, the cell is not able to further divide. So what it does is it undergoes death. This telomeric attrition, that means the stoppage of further cell divisions or making the cell die by the telomeres, is negated or is stopped by an enzyme called as telomerase. A telomerase is nothing but a DNA polymerase. DNA polymerase, in biochemistry, we know that any DNA polymerase, it will synthesize the DNA. So, okay, they will provide an RNA. Okay, they will provide a complementary substrate. In biochemistry, we are all aware that any DNA polymerase, they will synthesize the DNA. What does this telomerase do? The name itself says that it will produce a telomere again. That means it will elongate the telomeric DNA, and it will lead to uncontrolled division, and the cell is not going to die at all. How does this telomerase is produced? It is produced by the tumor cell. Okay, it sends signals, growth signals, and It may be secreted by the tumor cells, or it can be secreted by the paracrine signals, which will lead to a secretion of enhanced activity of telomerase. And it shortening of the telomeric end comes to an end. It elongates the telomeres, and it will lead to a replicative immortality. The cells will never die, or the cells is going to be live forever. So this is how occurs. The telomere is at the end of the chromosome. It gets shortened with every division. After one particular critical point is reached, there is going to be a loss of function. So what happens after this is that the cell cycle gets arrested. It either undergoes apoptosis, or the cell goes to senescence, or it can be a genomic instability. So what does this telomerase do? So this is the telomeric end. This telomerase is a DNA polymerase. It elongates the DNA of the telomere. So it ultimately leads to uncontrolled cell division, unstoppable cell division. So that's the name replicative immortality comes. Which of the following statements are true regarding telomerase? It has a DNA and a protein component. It is a DNA topoisomerase. It protects the integrity of chromosomal ends, and overexpression of telomerase is seen in all the cancer specimens, which is true. I'm getting an answer from only one person every time. Others can try. It's not going to be a subjective answer. It is going to be, you're going to say whether it is going to be option A, B, or C, or D. Can someone answer? I told you everything, no? It has both DNA and the protein component, of course. Is it a DNA topoisomerase? No, I told you it is a DNA polymerase. I told you now, no? I told you very clearly it is a DNA polymerase. It synthesizes DNA. It is not a DNA topoisomerase. Answer is B. It protects the, sorry, not answer is B. I asked for true statement, right? So it is also wrong. It has both, it protects the integrity of chromosomal ends. Of course, this is correct. It is not having a DNA and a protein component. It is having an RNA and a protein component. And overexpression of telomerase is seen in all the cancer specimens. It's not seen in absolutely all the cancer specimens. It's seen in most of the specimens, but not in all the specimens. Okay. The answer here is it protects the integrity of the chromosome. It's a very simple question. Answer is it protects the integrity. I asked for true option. So some words about the cancer stem cells. So along with the normal, along with the cancer cells, differentiated cancers. Once the cancer cells, they single cell they start division. They produce numerous cells, but all cells are not going to be alike. There are certain exceptions that we call it as a cancer stem cell. Okay, they are called as cancer initiating cells. Okay, what happens is that normally there are going to be a stem cell division. It will undergo differentiation and produce the progenitor cells, and these progenitor cells will undergo further division, which will lead to differentiated cells. What happens in a cancer stem cells? These cells are extraordinarily rare. That means 1,000 cells are there, 1,000 differentiated cells are there, only one cancer stem cells will be there. That means very, very, very rare within the tumor. So what we do is these differentiated cells, when we treat them, we can destroy them with either chemotherapy or anything else. Surgery is different. Surgery, we in total, we remove all the cells. We don't see it's a cancer stem cell or it's a differentiated cell or undifferentiated cell. But the other modalities of treatment, chemotherapy, radiotherapy, they destroy the usual differentiated cells. But these cancer stem cells, they survive. Okay, they usually make up less than 10% of the neoplastic cells, often less than 5%. The 95% of the cells are progenitor cells or the differentiated cells. Cancer stem cells, they can be able to generate tumors by self-removal. That is the difference between a differentiated cell and a cancer stem cell. As well as to differentiate into, it can get differentiated into multiple cell types. For example, okay, there are so many cancer cells are there, normal tumor cells are there. So there will be one cancer stem cell. Okay, these cancer stem cells, if you give a therapy, it's very easy to destroy all these other cells differentiated. Only if this escapes, it can produce a tumor again. This is a cause for recurrence usually, these cancer stem cells. It's a recent concept. Okay. Or it can give rise to a recurrence of the same tumor, or it can even produce multiple cell types. So it need not produce the same cell type. It can produce multiple cell types. Okay. So it can produce a different type of tumor. The same cells need not be produced again. That's what I wanted to insist. So when there are going to be more cancer stem cells, they are more resistant to standard therapies and are source of a tumor recurrence after achieving a initial remission. So first there is going to be a remission with a standard therapy, but if there is going to be a tumor recurrence, most of the time it is going to get initiated from the cancer stem cell. So this is the hypothesis. So oncogenic events occurs. This is a cancer stem cell. It produces progenitor cells and it produces different. These are all differentiated stem cells, and these are all progenitor cells. But reprogrammed cancer cell into one of the cancer cells may be reprogrammed to a cancer stem cell state. So conventional therapies will destroy all these cells. Once this cell is left out, tumor recurrence is going to occur. So angiogenesis, all of you are aware that usually I told you more than 1 millimeter. Already I told you the tumor cannot survive without a blood supply. So even if it is lesser than 1 millimeter, they will be able to survive in a stressed environment by the help of different processes. I told you autophagy is one of the processes, and here the angiogenesis, as far as it is concerned, the endothelial cells are very important in the formation of new blood vessels. These endothelial cells, they secrete various growth factors which are responsible for the synthesis of blood vessels. VEGF is one of the important factors. So basic and acidic fibroblast growth factor and platelet-derived growth factors are synthesized to enhance the neovascularization. The process of neovascularization is a little more elaborate. I didn't put it because we don't require so much to. The only thing is that you should know which factors are going to be angiogenic, and you can use angiogenesis inhibitors in certain tumors expressing this VEGF or PDGF depending on their activity. There was a question in NEET 2017. In the absence of blood supply, this line is taken from Bailey and Love. In the absence of blood supply, a mass of tumor cells cannot grow beyond the diameter of. I repeatedly told you. I don't think I need to wait for an answer. Everybody should be aware of it. So finally, how do they activate invasion and metastasis? Just remember that all plants will not grow in the same soil. Certain plants will grow in tropical, certain plants will grow in temperate. Certain plants will grow in South India, certain plants will grow in North India. The soil is different. So they choose selective environments, the plants. Similarly, the tumor cells also, they grow only in selective environment. For example, a CS stomach, gastric cancer cell can grow only in certain environment. It will not invade and metastasis to all the organs, but preferably can go to other organs. Sometimes a coconut tree can be grown in North India, but it will not grow in a nice way. Occasionally you can see, but likewise, it will not be. The tumor cells also, they require a selective environment. Other just tumor cells is the seed and soil is the environment. Okay, so when they have a good support for tumor growth, they grow well. Okay, that is called as a seed and soil hypothesis. So this seed and soil hypothesis are explained by different theories. First one is the growth factor theory. For example, you take liver or something, or brain. Okay, so liver is, if we take a transferrin as an example, in spite of carrier protein, though it's a carrier protein for iron, it also has some growth factor activity. This transferrin is excessive in liver or brain. So this, I'm just saying an example. Transferrin is excessive in liver. So what happens is that, so in breast cancer, the breast cancer cells, when they move in the blood circulation, this transferrin is a growth factor. This transferrin is a good growth factor for the breast cancer cell, not for any other cancer cell. So this breast cancer cell will preferably enter the liver, and they start growing as a metastasis. And in bone also, okay, bone also you have transferrin. So this is how growth factor theory explains only a certain tumor will metastasize and grow in only certain locations, not in all locations, preferably in certain locations. Okay, it can go anywhere, but preferably it can grow only in certain locations. That first theory, growth factor is transferring, so which attracts the breast cancer cells into the liver and bone, and it gives a good environment for the tumor growth. Next one is an adhesion theory. The endothelial cells, for example, you take in again in liver, you say so the blood vessels of the liver, the endothelial cells, they express certain adhesion molecules. Okay, these adhesion molecules, they selectively adhere certain cancer cells. Okay, and they lodge them inside the particular the same organ and make them grow. And third one is chemokines. So these certain chemokines which are secreted by target organ. For example, lungs. Okay, lungs, they secrete one factor called X. And one chemokine called X, and tumor cells, they come. Tumor cells, for example, you say breast cancer cells, they come in the blood circulation, and they have a receptor for this X. Okay, X receptor. So this specific chemokine can attract the tumor cells by just joining with the ligand, joining with the receptor in the specific cancer cells. For example, bone marrow, liver, and lungs, they express CXCL2 chemokine, which has ligand. This is the ligand which has a receptor CXCR4 in the breast cancer cells. So these three will selectively attract the breast cancer cells. That is why breast cancer is they commonly metastasize to the bone marrow, liver, and lung, not to elsewhere. Okay, so these are all theories. These are all the possible. Though the studies also have been done, and they indicate that these are all the possible ways certain locations they harbor certain type of cells. So metastasis. How does the metastasis occur? Why at all a tumor cell enter the why at all a tumor cell enter the blood circulation and go elsewhere, and they want to survive elsewhere also? See, for example, a emperor comes. First, he's a small king, and the emperor comes, and so his passion is to cover more place. He has to invade more place and bring everything into a small king. From a small king, he becomes a big emperor by invading so many places. Okay, like that. The tumor also, they want to dominate in the body. They want to show their significance to the body. So they want to invade wherever possible. They want to move around everywhere and try to lodge in and grow wherever there is a possible environment. So what happens here is that the formation of tumor metastasis is characterized by detachment of only certain tumor cells. Few tumor cells, they detach from the tumor mass, and from the primary tumor, they infiltrate into the bloodstream or lymphatic. This is called as intravasation. Okay, intravasation into the blood vessel, and the reciprocal process occurs at other locations. For example, from the breast through the lymphatic vessels. Okay, they intravasate into the lymphatic vessels. Intravasate into the lymphatic vessels. From the lymphatic, they go and extravasate to the lymph nodes. You understand? So that is how there is an intravasation followed by an extravasation. Oh, how does this extravasation occurs? I told you a lot of theories. Why selectively it occurs in, so though the blood flows everywhere in the body, and the tumor cells also flows in the blood everywhere. It goes to brain, it goes to bone, it goes to liver. Okay, so not in every tumor, not all the organs are affected, only certain organs are affected. Okay, it is mainly mediated by what is called as CAMs. CAMs is nothing but cell adhesion molecules. They adhere the cell. Okay, depending on the adhesions only, this cell and cell interaction, cell and matrix interactions, they are mediated by this cellular adhesion molecules. Example, cadherins, integrins, and selectins. How do these substances they help in these processes? I will tell you. We will take this one cadherin as an example. This is a prototype cadherin responsible for cell polarity and organization of the epithelium. So cadherin function is lost in most of the epithelial tumors during progression. So when there are going to be a proper function of a cadherin or integrin or selectin, these cadherins, they try to just make the tumor cells adhere to them. They will not allow the tumor cells to invade further. There is going to be a mutation or is a function loss of the E cadherin. This tumor will progress by invading the other places. So this is how the E cadherin functions. What happens is that there is going to see this E cadherin is attaching, this E cadherin bridges the two cells. Any cell has to live. It can be a normal cell or it can be a tumor cell also. If a tumor cell has to live, it has to cross the junction and go. Or if the tumor cell has to get detached from the tumor mass, this E cadherin, which is attaching the both cells, they should lose the function so that they will get detached from the tumor cells, tumor mass, and go to a circulation or elsewhere. So once the E cadherin function is lost, first of all, you should know the structure, how this E cadherin is. This E cadherin is attached to two cells are attached by E cadherin. This is mediated by what is called as a beta catenin and alpha catenin and actin. This is an apparatus. This is how a E cadherin is attached. What happens is that this E cadherin, when the E cadherin function is lost, this beta catenin, they get dislodged from the E cadherin receptor, and this beta catenin is present in the cytoplasm. When there is going to be excessive beta catenin, what happens is that this APC, adenomatous polyposis coli gene, is the one which phosphorylates the beta catenin and it degrades the beta catenin. This beta catenin is very notorious if it is going to be in the cytoplasm. Usually there is a loss of E cadherin function. This beta catenin gets released from here and it is present in the cytoplasm. Once it is not degraded by phosphorylation by the APC, when the APC is normal, it is not going to cause a tumor. But when the APC is also mutated, like in the case of a familial adenomatous polyposis, this beta catenin enters the nucleus and it promotes cellular proliferation by targeting specific genes. Okay, there is one more thing called WNT. Normally, this WNT signaling pathway is normally silent. Okay, sometimes in certain genes, this WNT signaling pathway may get activated and it inhibits the APC. So when it inhibits, so sometimes there may be a direct mutation of APC as in case of FAP, or sometimes there is normally the WNT is silent. So due to mutation, this WNT pathway gets activated and this WNT just suppresses the APC. So APC could not degrade the beta catenin. This beta catenin goes inside and causes the notorious effect. That's how it occurs in the. This is one of the proto model for carcinoma colon. These pathways are very much involved in carcinoma colon. This E cadherin, if you remember, E cadherin function is lost in diffuse gastric carcinoma. So Lawrence classification, if you remember, in the diffuse gastric carcinoma, this E cadherin function is lost. This is how it occurs. We just memorize E cadherin is in diffuse gastric carcinoma. Beta catenin function is lost in colorectal cancer. APC function is lost in colorectal cancer. Okay, like that. Beta catenin is increased in colorectal cancer. APC function is lost in colorectal cancer. We just memorize it. This is the pathway involved, and why it occurs, I explained you now. So we are all aware that any cancer can metastasize. Okay, how does it metastasize? I told there may be E cadherin loss so that the cell can get detached. So only it can get detached. How can it enter the blood circulation? How can it penetrate the endothelial cell? How can it travel in the blood circulation? How can it enter the other organs? Okay, just this is a very important process. The questions can be very much framed from this epithelial mesenchymal transition. This, though that carcinoma arises from epithelium. So epithelium are usually columnar or cuboidal, something like that. But so because of this epithelium, they may not be comfortable to enter the narrow spaces or just go through the bridges. What do they do is that they become like a mesenchymal. Mesenchymal cell. Mesenchymal cells are usually spindle shaped. Okay. This epithelial mesenchymal transition is very important in embryogenesis. In normal embryogenesis, because the cell has cells generated elsewhere has to get transported to elsewhere to produce the different organs in a normal fashion. This EMT is very important in normal embryogenesis also, as well in cancer. In the case of metastasis, this epithelial mesenchymal transition plays an important role. So it gets the paracrine signals from the macrophages, juxtacrine signals, myofibroblasts, and the other stroma cells also. These signals, what do they do? This just tells the cell, if you become a spindle shaped, only you can enter the blood circulation and go to a different place. So these cells, they undergo some processes and they become spindle shaped like a mesenchymal cell. Epithelial cell looks like a mesenchymal cell, and they enter the circulation. And how do they enter the circulation? There is going to be a signal from the platelets or any other blood cells. They just intravasate as a spindle cell, and they roam around as a spindle cell. And like something like epithelial mesenchymal transition, there occurs a mesenchymal epithelial transition when it enters the target organ. So here also it is spindle shaped, and it enters so that it can easily penetrate and enter the target organs. And once it reaches the target organs, it undergoes a mesenchymal epithelial transition and lodges in the organs and produces metastasis. Okay. Question: all the following are true regarding the epithelial mesenchymal transition except. Anyone can answer? Excellent. It is C. So epithelial mesenchymal transition is an essential phenomena during normal embryogenesis. I told you, cancer cells downregulate the expression of cellular addition molecules like E-cadherin. Yeah, that is important. Only if they downregulate the E-cadherin. Otherwise, E-cadherin will not allow you to detach from the cells. Okay, so this is important, and it is not essential for tumor formation. It is essential only for the tumor progression or invasion, not progression, tumor invasion and metastasis. Cancer cells, they use EMT to become invasive and metastasis. All others are correct. So it is not essential for tumor formation. Okay? It's only for invasion and metastasis. Okay, next is avoiding immune destruction. So to understand this, you should understand what is an immunosurveillance. In a cancer, in oncology, the main immunosurveillance, the main cell which is responsible for immune mediated destruction is T lymphocyte. Just remember that. Okay. So even in transplantation also it plays a role, but even in cancer, T lymphocyte is very important because it is specific for elimination of transformed cells, and there is a continuous surveillance for the transformed cells. So what happens is that there is going to be an abnormal cell, okay, that they call it as a cancer cell. So they recognize that, and the immune cells will infiltrate around the location, okay, with memory T cells. The memory T cells can be a CD4 T helper cells or a CD8 cytotoxic T cells. So T helper type 1 cells, right? So when there is going to be these kind of stuff present in the tumor mass, when you take a histopathology specimen and there are memory T cells present in the tumor mass, it is a good prognostic sign because it is prognostic for overall survival and disease specific survival in all cases of cancer. For example, in a stage 3 cancer, if the median survival is 2 years, when you take a HPE, when you take a true cut biopsy or a main specimen, and if there is going to be a lot of memory T cells present, that means the overall survival will be better. If the T cells are very less, the overall survival will be even less than 2 years. If the T cells are present, it will be more than 2 years. Like that, we can just, for the understanding sake, I'm saying, when there are going to be memory T cells in the tumor mass, there is a possibility of a better survival. Alternatively, if the myeloid cells are present, it correlates a worse prognosis in cancers like pancreas and blood cancer. What is immunoediting? So the immune system, they put on a continuous pressure when the tumor is evolving. Okay, so they play a dual role, okay, between a tumor and the host. So what what is the first role? It effectively eliminates highly immunogenic tumor cells. The first role is a protective role. So when there is an abnormal cell, tumor cell, the immunogenic cells, okay, they go and first of all, this tumor cells should be immunogenic. Okay, when it is immunogenic, that means the cells will induce the immune cells to come to the site so that the immune cells will do their action by effectively eliminating the tumor cells. This is the protective. At the same time, when the tumor cells, they disguise themselves. They are tumor cells. For example, a T cell comes and asks, hey, why do you come? You are a tumor cell. You are not a normal cell. Why do you come now and get lodged in the normal environment? He says, I am not a tumor cell. I am a normal cell. They will disguise themselves like a normal cell so that the immune system, they fail to recognize that the tumor cell is the tumor cell itself. And they think that it is going to be a normal cell, and they are not going to destroy the tumor cells. So that means we say that they evade the immune system. That is what the point. They avoid immune destruction and they evade the immune system. How do they do that? Okay. So for example, you see here, this is a normal tissue. Because of the tumor suppression, senescent apoptosis and all, these normal cells, when they produce tumor cells, they come back to normal. That is because when the intrinsic tumor suppression processes are normal, they revert back to normal. So suppose there are carcinogens or radiations or any other carcinogen exposure, what happens is that these cells will this will lead to a transformed cell. This transformed cell is usually a highly immunogenic cell. That means these cells will be recognized nicely by the T lymphocytes and other immune cells. So CD8 cells, CD4 cells, okay, and natural killer cells, all these stuff will come in, and they will, because they are immunogenic, they show themselves as a foreign cell, they show themselves as an abnormal cell. These cells will come and destroy the transformed cells and lead to what is called as an elimination. What happens? Elimination means though there is a transformed cell produced, our body mechanisms, our immune mechanisms, they destroy the cell and they revert back to the normal stage. That is called as elimination. After certain stage, what happens is that these cells, they disguise themselves. That is, disguise means they act themselves as a poorly immunogenic cell. They will not show that they are tumor cells. For example, this purple and this brown cells, they are all transformed cells but with poor immunogenicity. They are not recognized by our immune cells, CD8 and CD4. They don't recognize these cells. So they recognize the other cells, immunogenic cells, and they destroy only them, and they will reach an equilibrium. After that, then antigen and major histocompatibility losses occur, and it will lead to escape from the normal protective mechanisms, and it will lead to a tumor growth promotion. So how does this escape? So we say that there is a lack of immunogenicity. Usually I will tell one or two examples. How does these tumor cells escape the elimination phase? By showing less immunogenic show. So just remember there are two important, this is very important in the immunotherapy and immune processes. That is a separate topic by itself, a separate chapter in subdiscipline. But I'm not going to be going in depth about that. Just remember that normally the CD86 from the antigen presenting cell, this is okay. Normally this is attached to a CD28, okay, and for the normal T lymphocyte to function, okay, this will go and attack the antigen and it will destroy the antigen. What happens is that the same substance can be attached to a CTLA4, cytotoxic T lymphocyte antigen 4. Okay, this CTLA4 can attach themselves to the CD80-86. This attachment, what happens is that this attachment, which will reduce the signal given by the CD80, CD28, and it will this will not enhance the immunity of that antigen to the lymphocyte. The T lymphocyte will, it will be cheated by the, because the CTLA4 just downregulates the antigen presenting cell, and this antigen will not be recognized and this will not be destroyed. This is how the cancer cells, they reduce the immunogenicity. Another one is that there is a substance called PD1, okay, programmed death 1, and it has a ligand in the cancer cell, PDL1. Okay, PDL1 is the programmed death ligand 1. So what happens is that once this PDL1 attaches with the PD1, programmed death 1, it sends inhibitory signals, and these lymphocytes will not be able to understand what is going on. There will not be a good immuno recognition, and the T lymphocyte will not be able to identify the cancer cell and destroy it. So these are all the targets for therapy. So these are all targeted therapies. We can avoid the combination of PD1 and PDL1 by giving an anti-PD1 or anti-PDL1. Anti-PD1 inhibitors like pembrolizumab can be used. Anti-CTLA4 inhibitors like ipilimumab. These are all highly, these are all very recent drugs and very impressive names for the examiners to ask this question. So, which of the following is a CTLA4 inhibitor? Is ipilimumab, and anti-PD1 inhibitor is pembrolizumab. Dysregulating cellular genetics. Just we have to understand what is a Warburg effect. Okay, so what happens in a Warburg effect? So dysregulating cellular energetics in the sense, though the tumor cells, they just they have a predilection. They favor energy metabolism to glycolysis even in the presence of oxygen. Okay, so what happens is that this glycolysis usually, as we know from the biochemistry, it yields only 18-fold lower energy yield when compared with aerobic metabolism. Hence, it has to upregulate the rate of glycolysis to 200-fold. So it will not undergo a mitochondrial aerobic metabolism for the energy production. So these tumor cells are abnormal cells. They behave in an abnormal way. Normal cells, they undergo aerobic metabolism, aerobic metabolism to produce energy. But here, this, though you have all these stuffs available, they prefer glycolysis. They like only glycolysis. And in the glycolysis, during this time, what happens is that it just upregulates the expression of glucose transporters like GLUT1. Glucose transporter 1 is upregulated. So this Warburg effect is the concept used in the PET-CT. When you use an 18-fluorodeoxyglucose, this is selectively taken by the tumor cells because they undergo only glycolysis and they upregulate the expression of GLUT1. So this FTG are selectively taken by the tumor cells, and it is very much used nowadays. This nuclear medicine studies like PETs are used very much nowadays to diagnose and stage the cancer. Finally, next is genomic instability and mutation. Under normal physiologic conditions, genome is maintained in an extraordinary fitness because of the caretaker genes, especially P53, retinoblastoma, etc. So what is going to happen is that is going to be a alterations in the caretaker genes. Okay, what happens here? Loss of ability to detect the DNA damage. P53 is normal, wild type P53, they will be able to recognize the DNA damage. When the P53 is mutated, they are not able to recognize the DNA damage and they will not send the cell for apoptosis. So the further proliferation occurs. Loss of the ability to directly repair the damaged DNA. Okay, they are not able to repair the damaged DNA. Though they recognize this, they may not be able to repair it. And inability to inactivate and intercept mutagenic molecules. So they are not able to inactivate the mutagenic molecules before the DNA damage can occur. Ideally, if they are maintained with extraordinary fidelity, they will not allow the mutagenic molecules to come near the damage itself, come near the DNA itself. So here, then there is going to be a alteration in the caretaker genes like P53. These stuffs cannot be maintained. They lose all these abilities and finally lead to a mutation, which the mutation leads to a uncontrolled proliferation. P53 is the most commonly mutated. So first tumor suppressor gene to be cloned is retinoblastoma gene. P53 is the most commonly mutated tumor suppressor gene. Okay, questions can be framed in that. So there can be a question asked in the Warburg effect also. So which effect is utilized by the PET scan in staging and diagnosing the cancers? There can be 4 options given. Warburg effect may be one of the options, and you can choose the Warburg effect. Tumor promoting inflammation, I told you in necrosis itself. So when there is going to be a inflammation, okay, there are going to be even though the immune cells, they try to destroy the tumor cells, in paradoxically sometimes the same immune cells, they can also enhance the tumor progression. We have seen this already. Leukocytes, they produce various growth factors which can promote the tumor growth, angiogenesis, and therapeutic resistance. This we have seen already. That is about the main tumor biology and the processes which usually occurs in the tumor initiation, tumor progression, invasion, and metastasis. I think I have cleared some of the concepts, and these concepts, I don't know how far you can take home so many messages, but the certain logic, certain concepts will help you to easily crack certain questions asked from molecular biology. You can easily rule out the options if you understand certain facts. And moreover, this is going to be helpful when you approach the individual cancers. When you study for the entrance exam, you will be approaching breast cancer, CA colon, CA stomach, and all. When you approach individual cancer, these concepts, it will really help you, and it will save your time to understand. And you can revise many times. You can save the time, and you can easily approach those topics. So next, I will spend few more time in cancer genetics, maybe for another half an hour or so, and we will wind up the session after that. Cancer genetics. So we all know that all malignant transformations, they occur at a genetic level. It can be a gain of function mutation of oncogenes or loss of function mutation of the tumor suppressor genes. So mutations, how does that occur? It can be a germline mutation, or it can be a somatic mutation. What do you mean by a germline mutation or a constitutional mutation? It is inherited from one of the parents. Okay, it can be inherited from both the parents, or it is inherited from one generation to the other. And once it is inherited, it is present in all the cells of the body. So in case of a germline mutation, what happens is a hereditary cancer occurs. When in a hereditary cancer, it usually manifests in an early age, and the tumors are going to be multifocal, bilateral, and highly aggressive when compared with the sporadic cancers which are going to occur due to a somatic mutation. Somatic mutations, I told you, they are acquired mutations. Okay, this is not transmitted from one's parents. No, it is acquired during individual's lifetime, and it is acquired due to different processes. It is acquired mainly due to environmental processes, or it is acquired due to lifestyle changes. That is what the environmental processes, and it is not going to be passed to the next generation, and it is not going to be present in all the cells of the body. Germline, see, one cell from father, one cell from the female. Okay, zygote is formed. Okay, diploid zygote is formed. So that cell has this germline mutation. All the further divisions are going to happen from the same cell. So this mutation is going to present in all the cells of the body, germline mutation. But somatic mutation, due to an environmental, for example, a smoker has an influence in the smoking has influence in the lung. Okay, sorry. Okay, so what happens? There is going to be a change of one particular cell due to the environmental carcinogen. The cell divides, and it is going to divide and form a tumor. That mutations are going to be present only in that tumor cell. Okay, if it metastasizes, wherever it goes, then all the other cells also will have the but in case of germline mutation, that mutation will be present all the cells of the body. Somatic mutations, wherever that is mutated, only that place it is present. So what do you mean by an index case or a proband? Index case, in a germline mutation, there are going to be, in case the tumors or tumor syndromes caused by germline mutations, it's going to manifest in the individual at a particular age. Okay, so they have a syndrome. It is the first individual who is going to be diagnosed with the syndrome. They are called as index case. Even if the earlier generations or the previous generations are affected, if they are recognized only after this patient, the index case is the first individual who is diagnosed with the syndrome, even though it may be present in the previous generations. So germline mutations, somatic mutations, are they different? So hereditary cancers or sporadic cancers, are the is it going to be different mutations in the same gene? No, not going to be a different mutation. Most of the time, they are often the same as the somatic mutations. Germline mutations and somatic mutations, they are most commonly alike. For example, familial adenomatous polyposis caused by a APC gene mutation. More than 80% of the sporadic colorectal cancers also have a same somatic mutation of the same gene. Even though it is sporadic, the same gene is affected in 80% of the patients. But in case of a germline mutation in case of a hereditary cancer syndrome, it is 100%. So a person with hereditary cancer syndrome, 100% that mutation will be present. Then only we can call it as a hereditary. But same cancer can occur sporadically. Most often it occurs sporadically. All the cancers, for example, colon cancer, if you say sporadic breast cancer, you say 90% of the time it is going to be sporadic only. Only 5 to 10% of the times it is going to be hereditary. Okay, so even though it is sporadic or hereditary, even though the hereditary syndromes, this 5 to 10% will have 100% of these patients will have a particular mutation for that syndrome. In sporadic also, the same mutation may be present or a different mutation also may be present. So I'm saying that many a times, often hereditary mutations and sporadic mutations are alike. So most of the familial cancer syndromes are autosomally dominant. They are inherited in an autosomal dominant fashion, except ataxia telangiectasia and xeroderma pigmentosa. They are inherited in an autosomal recessive fashion. So this is a question. All are inherited by autosomal dominant means, except 4 options can be given. FAP, BRCA, and CDH1 and ataxia telangiectasia. You have to choose ataxia telangiectasia. They are inherited in an autosomal recessive fashion. Not all the inherited genetic mutations have a complete penetrance. What do you mean by that? For example, there is going to be a APC mutation in 100% we say. Okay, so not all 100% will develop. So don't take APC, but I'm saying that any BRCA also, you say BRCA1, BRCA1. If 100 people are affected, 100 patients, they will not develop CA breast or CA ovary. Only about 60 to 90% of the patients. That is, out of that, maximum 60 patients or 90 patients only will develop a CA breast. Not all the patients will develop a CA breast. Okay, that means they won't have a complete penetrance. Even though there is going to be a hereditary, the mother transmits the mutated gene, goes to the daughters. But even though they transmit, not all daughters need to be developing a carcinoma breast. About 5 to 10 daughters, out of 100, they may escape the carcinoma. But we don't know who will develop and who will not develop. So we have to take a prophylactic treatment for all the 100 patients. There is almost complete penetrance of colorectal cancer in FAP. I told there are some exceptions. Almost 100% of the patients. I told in breast cancer it is 90%. In FAP, in familial adenomatous polyposis, there is going to be almost 100% penetrance. And almost, if there is going to be 100 patients with APC mutation, almost 100, all the 100 will develop colorectal cancer. And also in MEN2, RET mutation, RET mutation, oncogene mutation, in MEN2 syndrome, multiple endocrine neoplasia syndrome, medullary thyroid carcinoma occurs in 100% of the individuals. But MEN2 syndrome will have other cancers also. But other cancers, they are variable. For example, pheochromocytoma, they manifest only in 50% of the individuals, though they have a mutation. But MEN2 mutation, the defining identity is the medullary thyroid carcinoma, which will be developing in 100% of the individuals. That's why in a RET mutation, MEN2B syndrome, you have to undergo N2A and MEN2B syndrome. You have to make the patient undergo a prophylactic thyroidectomy. In contrast, here I told it's almost complete penetration. In contrast, less than 50% for pheochromocytoma in neurofibromatosis. Not all, only 50 patients out of 100 patients with neurofibromatosis will develop pheochromocytoma. So penetrance is variable. You just understand that. Not always there is a complete penetrance. This is a question for you. Mr. Ramesh had a daughter and a son. A month back, Mr. Ramesh's daughter was diagnosed to have a FAP. After that, further screening revealed the same mutations in Mr. Ramesh, his son, and his father. So all of them had the same mutation. Here, the index case. Who is the index case? Anyone can answer? You understood what is an index case, right? Excellent. Ramesh's daughter. So who was first identified? Even though previous generations are affected, Mr. Ramesh is affected, Mr. Ramesh's father, that means Ramesh's father is affected, the previous generation, the generation and the further generation is affected. The first case who was diagnosed was his daughter. So the index case or proband is Ramesh's daughter. So here is the genetic model. In the first slide itself, I told you that it is going to be a multi-step process involving activation of the oncogenes and suppression of the tumor suppressor genes. Okay, activation of the oncogenes or suppression of the tumor suppressor genes, which leads to a carcinoma and metastasis. We take this genetic model of a colorectal tumorigenesis. So normal epithelium, because of APC mutation in the 5q gene, it becomes a hyperproliferative epithelium. And due to DNA hypomethylation, I'll tell you about hypomethylation later. The early adenoma occurs, and KRAS mutation, oncogene mutation, you get an intermediate adenoma. DCC tumor suppressor mutation, you get a late adenoma. And P53 is a very late event. Okay, it occurs in the conversion of carcinoma. Late adenoma becomes a carcinoma. So there may be a question, which gene is responsible for the early in the cyclical change of colorectal tumorigenesis? 4 options can be given: APC, KRAS, DCC, P53. The answer will be APC. Understand? Earlier in the process, APC is involved, 5q mutation or APC mutation. Late, which gene is responsible for the late in the process of colorectal tumorigenesis? Or there may be a question like, which gene is responsible for conversion of a late adenoma to a carcinoma? In that case, answer is P53. So some few facts about the familial cancer syndrome is almost, this will help you when you go for the individual cancers. Only some important highlights. It will be a revision kind when you go to an individual. Not hardly another 10 minutes I'll go with that. And following that, some carcinogens, we'll wind up with another 20-25 minutes. So retinoblastoma, I told it's the first gene to be cloned, first tumor suppressor gene to be cloned. Most of the cases are identified at the age of 7 years, but bilateral disease, they are manifested even in the first year of life. Extraocular malignant neoplasms, they are notorious to cause sarcomas, especially osteosarcomas. Okay, that's just remember, if there is going to be in retinoblastoma, which cancer is caused, and there are 4 options, and there is no retinoblastoma, you have an option like osteosarcoma, answer is osteosarcoma, and sometimes they can lead to melanomas and CNS tumors also. So when you think about the tumor suppressor gene, when you discuss about the tumor suppressor gene, we must be aware of the nuisance to hit hypothesis. It applies to most of the tumor suppressor genes. Okay, most of the syndromes caused by tumor suppressor genes. So we take retinoblastoma as an example. Okay. First, they are heterozygous. Okay. In the next generation, again they are heterozygous. Okay. So the gene is carried by the first generation, it is transmitted to the next generation. Okay, so not everybody will develop a everybody will develop a cancer. Only when that becomes homozygous, that means first it is given by the hereditary itself. The father itself gives a defective gene to a next generation, and they just expand the single abnormal gene to the further generations. But it doesn't occur with the cancer doesn't manifest in all the individuals. They require a second hit due to an environmental factor. So just because the gene is sent and it is heterozygous, it will not manifest the syndrome, manifest the cancer. Only when there is going to be a second hit due to a environmental effect, that means the somatic mutation, then only the tumor is going to manifest. Otherwise, if there is now not going to be a second hit, okay, for example, if the retinoblastoma gene is inherited to the next generation, okay, this is the next generation. If there is going to be a cancer, if the cancer is going to arise, it should arise immediately, right? Maybe within few years, within 1 year or within 2 years. It's not going to be occurring so. It is going to be occurring in a later age, and it is not going to be the same age in which all the patients are going to manifest. It is going to manifest in different ages in different patients. That means that requires a two hits. One hit is given by hereditary, and next hit is given by the environment. And the tumor will manifest once there is going to be a second hit. Sometimes there is no hereditary defect itself. So one generation undergoes a non-hereditary environmental defect, but that defect goes to the next generation, and the next generation the second hit occurs and the tumor manifests. Understand that? So first hit is not enough. It requires a homozygous transmission for a tumor suppressor gene to manifest. So it applies for all the tumor suppressor genes. Okay, there may be a question. Okay, grandfather is Mr. X is affected with retinoblastoma, followed by his son and the daughter are not affected, but his grandchildren are again affected by retinoblastoma. That means the intermediate generation is a carrier for the gene. So that means that they may ask a question like this. It indicates what type of hypothesis, and they can give 4 options. One may be Knudsen's hypothesis. You have to go for Knudsen's hypothesis. Life harmony, you all are aware that it is due to a mutation in p53 gene in 50% of the patients only. And what are the carcinomas what are the cancers which can occur is sarcomas, both soft tissue and bone sarcomas can occur, breast cancers, brain tumors, leukemias, adenocortical carcinomas, and variety of other cancers can occur. You are all aware of the life harmony. The gene affected is p53, and the most common cancer which occurs in the life harmony syndrome is breast cancer. Okay, and what only in 50% of the patients, directly there is mutation in the p53 gene, but in other patients, rest of the patients, there are mutations in the different places, like checkpoint kinases like CHK1 and CHK2. Just remember that they are checkpoints for TP53. They phosphorylate the P53. So it is, though direct mutation is present only in 50%, it is revolving around the P53 only. If in the other 50% of the patients, the other genes which are affected are going to directly or indirectly involve the P53 deactivation. That is how the life harmony syndrome occurs. So life harmony syndrome, there may be a question like this. In life harmony syndrome, the gene mutated will be the question, and 4 options may be given. P53 may not be one option. There may be an option CHK2 and 3 more options. In that case, when there is no P53 mutation is given in the option, you have to go for a CHK2 or CHK1 mutation. How do you define life harmony? Proband diagnosed with sarcoma before 45 years of age, along with a first degree relative with any cancer. Any cancer in the sense the cancer pertinent to the life harmony syndrome, either brain tumor, leukemia, or breast. diagnosed before the age of 45 years, and plus an additional first or second degree relative with either a sarcoma at any age or any cancer before the age of 45. Any cancer means any cancer which comes under life-Romney syndrome, not any cancer which is out of the life-Romney. So this is the definition. When it is present only, you can diagnose it as a life-Romney syndrome. So which is the most common cancer associated with life-Romney syndrome? All these tumors are associated with life-Romney, which is the most common cancer. It was asked in one of the DNB super specialty. Just now I told you, no, you are saying sarcoma. When I was explaining this slide, I told you, and I just marked also. See here. Yes, it is breast cancer. Excellent. Next is FAP. FAP contributes only 1% of colorectal tumor burden. So 5q21 is affected. I told you APC mutations occur early in the adenoma carcinoma sequence. Okay, that is called as adenoma carcinoma sequence. APC is a early change that occurs in early adenoma. Later part P53. Okay, just remember that. Penetrance is greater than 90%. It is 100%. I told you already. If 100 patients are having a mutation in APC, almost 100 patients will develop colorectal carcinoma. Usually it occurs in second or third decade. The phenotype, the manifestation, colorectal cancer manifests in second or third decade. Okay, that means second decade in the sense 10 to 30 years. Very early it is. Okay, untreated individuals, they typically present with colorectal carcinoma at 35 to 40 years of age. That means untreated individuals in the sense, what is the treatment you can give? You can do a profile, like, because if it is present 100%, the patient is going to develop a colorectal carcinoma, and due to complications he is going to die. So what treatment you can give? You have to knock off all possible potential sites. That means it's a total proctocolectomy is the ideal option for total prophylactic total proctocolectomy is the ideal option for FAP individuals. So if you are not going to treat the patient, you have to, mostly they will develop tumor before 35 to 40 years of age. So when compared with sporadic CRC, I told it is going to be only 1% of the burden. That means 100 cases of CRC develop, only one case is due to FAP. Okay, rest of the cases are due to some other syndrome or sporadic. Most common is sporadic. 90% is sporadic. Most of the cancers, breast cancer, colorectal cancer, any cancer, it's mostly environmental change. It's sporadic. So this is only 1%. Okay. So when compared with sporadic colorectal cancer, this age is approximately 30 years earlier than the median age. So usually that means 65 to 70 years only sporadic colorectal carcinoma will manifest. But in case of a FAP, if you are not going to treat the patient, it is going to manifest in 35 to 40 years, maximum 35 to 40 years. Extracolonic manifestations, you have upper GI polyps, desmoid tumors in 15%, and thyroid cancers usually are papillary cancer. So which type of thyroid cancer occurs in FAP may be one question, and 4 options may be given, like Hurtl cell, follicular, papillary, like that. You have to go for papillary. So polyps of the stomach and duodenum are present in more than 90% of the patients. So stomach and duodenal polyps are present, and in duodenum specifically, most of the patients, they have polyps located in the periampullary region. If this polyps is going to get converted to a carcinoma, it may lead to a periampullary carcinoma from the duodenum. So when there is going to be a periampullary carcinoma arising from the duodenal cells, if you are diagnosing the patient with that, you have to screen for FAP, because most of the time it is going to be present. Even though you treat the duodenal polyps, other tumors may come. So it's going to be a periampullary carcinoma due to a duodenal carcinoma. You have to look for FAP. Indeed, duodenal adenocarcinoma is the third leading cause of death in FAP after metastatic colorectal carcinoma and desmoid tumors. These are all words taken exactly from the Sabiston latest edition, 20th edition. So there may be a question, After metastatic colorectal carcinoma, which is the commonest cause of death in familial adenomatous polyposis? What is the answer? After metastatic colorectal carcinoma, which is the commonest cause for death in FAP? Can anyone write in chat box? It's desmoid tumors. I have given it. So this is the classic FAP we have been talking about, and there are variations, okay, from the normal classical FAP. So you have mutations, like that is going. It's not going to be a single location mutation. There is going to be a range of mutations. Okay, from the 5'N to 3'N, you have in DNA, in the gene, it is going to be a mutation occurring at different sites within one segment, depending upon the location, whether it is 1043 or 2020, like that. You have different locations. Depending upon the locations, the manifestations will differ. For a classic FAP, you have a different location, and the variations are due to the location of the mutation in different sites. In Gardner syndrome, the location is different. In addition to colorectal carcinoma, osteomas of the mandible and skull will be present. Epidermal cysts, multiple skin and soft tissue lesions will be present, and especially desmoids and thyroid tumors will be present. So there is something called, that's a classical FAP. There is something called as attenuated FAP. This is very important. Okay, MCQ point of view, it is very important, right? So it is complete. It is not exactly similar to classical FAP. That here also the location of the mutation is different. I told you, the number. So in this segment, mutation occurs, it's going to be classical. Next segment, but all are neighboring regions only. Depending on the location that is given, all the locations are given where it occurs in Gardner syndrome, where it occurs in attenuated FAP, where it occurs in classical FAP. Everything is given in the text. But I don't want to further proceed with that. I don't think the questions can be framed from that. Just have a concept that depending on the location of the mutation, the phenotype will be decided. In Gardner syndrome, these are all the phenotypes. And in attenuated FAP, it is completely distinct. It is completely different from FAP. In that, usually in classic FAP, you will have more than 100 adenomas. That is one of the diagnostic feature of classic FAP. But in attenuated FAP, you have fewer than 100 adenomas. The polyps are more proximally distributed in the colon. The onset of colorectal cancer is about 15 years later. And there we told second or third decade. From that age, it is almost 15 years later in the patients with classic FAP. So in attenuated FAP, less than 100, proximally distributed, and it manifests 15 years later than the patients with classical FAP. Mutation responses for variant occur in the extreme upstream or downstream of portions of APCJ. I told you, location of the mutations will be extremely upstream or extremely downstream. So classic FAP is classical. In classic FAP, the mutation is in the center. Attenuated FAP differs from classical FAP by all except. Anyone can answer? Can you answer, please? Anyone can answer? I'm waiting for at least one answer. Dr. Roshni, it is. I told that in attenuated FAP, it occurs 15 years later than. That was the point given. This is the previous slide one. I told in attenuated FAP, you have proximally distributed cancers in the colon. Okay. So this option has distally distributed. So all other options are correct. Answer is polyps are more proximally distributed, not distally distributed. I told you except. MYH associated process, the MUT-Y homolog or MYH gene, which encodes a DNA glycosylase involved in base excision repair pathway. So all other either attenuated FAP, Gardner syndrome, classical FAP, and all other stuff are autosomal dominant. This MYH associated polymorphosis is autosomal recessive. Okay. Just remember that only this is the exception. It accounts for only a third of patients who have attenuated polyposis. Here, the phenotype is almost similar to the attenuated polyposis, but they test negative for APC mutation. In an attenuated polyposis, due to APC mutation, that is different. In MIH, the phenotype is similar, but they test negative for APC mutation. The polyps are distributed throughout the colon. Proximal distal predominance is not studied properly. They say that it's distributed throughout the colon. But in case of an attenuated FAP, you have proximally distributed tumor. Next is the Lynch syndrome. It constitutes only 2% of the colorectal. Mismatch repair genes you should be aware of. MSH2 and MLH1 contributes to more than 90% of the mutations. Other mutations in PMS1, MSH6 can occur. Penetrance is 80%. That means out of 100% positive, only 80 patients will develop the syndrome. So it has two types. Type 1 Lynch syndrome is they develop only colorectal carcinoma. In type 2, colorectal carcinoma with extracolonic carcinoma. More commonly, endometrium is the next most common cancer which is associated with Lynch syndrome. Lynch type 2, colorectal plus extracolon. Type 1, you have only colorectal. So right-sided predominance of colonic cancers, 70% are proximal to the splenic flexure. In case of HNPCC, most of the tumors, they develop proximal to the splenic flexure and they appear at an early age, median age of 45 years. I told in case of FAP, it is manifested still more earlier, second and third decade. I told it is between 10 and 30, but this occurs at a little later stage of about 45 years with increased likelihood of synchronous and metachronous cancer. So when they manifest, they need not have a cancer at one particular site alone. They may have a synchronous cancers, means when you identify itself, that time itself you will have more than one cancer. And
एक मिनट होल्ड कीजिएगा अरुण सर, डॉक्टर अरुण सर, it's not audible.
Mutations of the genes. Okay, so if there is going to be a methylation process which is going to occur in promoter CGIs, it is associated with closed chromatin structure, and there is a closed chromatin structure and transcriptional silencing of the associated gene. So it has shown to be a common. Yes. What happened?
Sir, you can continue.
Okay. So it has been a common event in carcinogenesis. So just remember one fact, normally it is unmethylated. Okay. So methylation occurs due to an abnormal process of these CpG islands. and it relates to a closed chromatin structure and transcriptional silencing of the associated gene. So what is the associated gene? These genes like tumor suppressor genes like CDKN2A. CDKN2A is, there is going to be a mutation in CDKN2A. You can develop a melanoma or a familial melanoma or familial pancreatic cancer. And retinoblastoma or von Hippel-Lindau or BRCA1. These are all inactivated by the hypermethylation of the promoter CpG islands. Okay, just remember, is an epigenetic material. That means it is not in the gene, but it is present close to the gene. Epi means it is over the gene or it is close to the gene. These CpG islands, they are normally in normal cells in an unmethylated state, and they are present located close to the 5' ends of the genes. And this methylation occurs due to some abnormal processes, and it leads to a mutation in these tumor suppressor genes, and respective carcinomas can occur. It's not moving.
Put a escape button. So yeah, it's moving.
Now it's moving. Yeah. So it is not always that unmethylated states are present in normal cells. In certain places, certain other epigenetic materials, they may be in CpG, may be in hypomethylated state. For example, promoted CpG demethylation has been shown to lead to overexpression. So I told in this retinoblastoma, BRCA, breast cancer, and melanoma, usually hypermethylation occurs of the CpG occurs, which mutates these genes. But here in gastric cancer, what happens is that demethylation occurs. Normally it is methylated, okay, but as a pathological process, demethylation occurs, and it causes overexpression of the cyclin D2. Cyclin D2 is nothing but the oncogene. The cyclin D2 has a role in the G1 phase of the cell cycle. When it is going to be excessively expressed, it is going to cause an uncontrolled proliferation. And there is one more product called maspin in gastric cancer. So it's not only the hypermethylation, it's also sometimes demethylation, which leads to certain type of cancers. Okay, but hypermethylation occurs and causes mutation in tumor suppressor genes, but demethylation occurs and causes overexpression of oncogenes. Just remember that. This is how it is. This is an unmethylated CpG island. Okay, so once there is going to be an hypermethylation due to a transcriptional repressors or histone deacetylases or other stuffs. See this blackish one, they are all I mean hypermethylated, and here the transcription is abolished and it leads to cancer. Genomic imprinting. So next, so it's one more type of epigenetic way of causing cancer. Here, conditioning of the maternal and paternal genomes, they occur during the gametogenesis itself, such that a specific parental allele is more abundantly expressed in the offspring. For example, a maternal or a paternal, exclusively most of the genomes are derived from one particular parent. For example, in Wilms tumors, this loss of imprinting has been demonstrated, leading to a pathological expression of IGF-2. I told insulin and insulin-like growth factors, when there is going to be excessive expression, they lead to tumor, they lead to cancer. So there is also one more change. It occurs along with the biallelic expression of IGF-2. Like this, there is a hypermethylation of regions of reciprocally imprinted H19 gene. These are all you have to keep it in mind only. So it's very difficult to give description for all these stuffs put up. Otherwise, I think that's not required also. Just remember, genomic imprinting is a type of epigenetic modification leading to cancers. So here what happens is that one of the parents' genome is exclusively expressed in the offspring, which leads to, that means if the father is having a particular mutation in the particular gene, any defect in the epigenetic part, and this what happens is that exclusively the father's genome is going to be expressed. So what happens is that you have a biallelic expression of some particular abnormality in that particular cancer occurs. These two phenomena are earliest detectable changes in this Wilms cancer. They strongly suggest that they have a gatekeeper role in these alterations. The last one in cancer epigenetics is modification of histones. It can occur then. The histones are also epigenetic materials. Depending on their modification, there may be a stimulus which leads to a cancer pathway, which leads to cell proliferation. It can occur by acetylation, methylation, phosphorylation. In CpG islands, it's only the methylation, hypermethylation or hypomethylation. Okay, here histones can be modified by acetylation, methylation, phosphorylation, and is important in compaction of the chromatin structure. That means they constrict the chromatin structure. Recent work in colorectal cancer, they suggest that DNA hypermethylation together with histone modifications play a critical role in maintenance of gene silencing. This is the activity you see here. This is H, okay, histone deacetylase. What happens is hyperacetylation of substrates occurs. So histones, they modulate DNA methylations, DNA methylation or acetylation or any other process occur, they modulate, they increase the cellular stress, and either a cell death occurs or survival and differentiation occurs. Okay, this is so. So if you want to stop this process, you can use a histone deacetylase inhibitors. So just few more slides about the carcinogens. I'm not going to be telling in detail about each and individual carcinogen and what is the cancer it is going to cause. I'll just highlight some concepts and how it is divided, and I will just tell you where to read all these stuffs. Okay. So carcinogens are commonly divided into 5 groups depending upon their efficiency of causing cancer. Okay. So group 1, 2A, 2B, 3, 4. There is no group 5. Though it is 5 groups, it is only group 1, and 2 has 2A and 2B, and group 3 and group 4. Group 1, they are all proven human carcinogens. Probable human carcinogens for which there is limited evidence of carcinogenicity in humans, but there is sufficient evidence to prove carcinogenicity in experimental animals. That means in animals there is a sufficient evidence, but in humans there is only limited evidence. That is 2A. 2B, they are probably carcinogenic. Okay, probably means most of the time they are carcinogenic. Here they are possibly carcinogenic. Just make the difference. Possibly means occasionally, rarely, maybe, like that. Probably means mostly, possibly, maybe. Okay, carcinogenic for humans, but limited evidence in humans and less than sufficient evidence in animals. Group 3, you have inadequate evidence for in humans. There is no proper evidence, but in animals, in both humans and animals, there is only inadequate evidence. Group 4, they are not carcinogenic probably. Okay. So this is these are all the tables given in all the textbooks. You can go through. This is a table I put it from the Sabiston for 20th edition, and you can have it in Devita also. You can also have it in other oncology books. This is going to be the same. There may be questions asked from these kind of tables, particularly in NEET super specialty oncology exams. Mostly they prefer taking questions from tables of Sabiston or Bailey. So just go through this table. Okay, it is in the chapter 28 of Sabiston. These are all pharmaceutical carcinogens. You know, tamoxifen, though it is a treatment for CA breast, it can cause endometrial cancer, and estrogens, breast and endometrium can occur. So you have other stuff, azathioprine, non-Hodgkin's, cyclophosphamide, though it's a chemotherapeutic drug, urinary bladder and leukemia can occur. So infectious carcinogens, you will be aware of most of the stuffs. Okay, papillomavirus, you know, cervical cancer, anal cancer, H. pylori, cystosoma, everything you will be aware of. So tell me, the environmental agent X is proven carcinogen to is a proven carcinogen to human. X belongs to which group? Anyone has replied? I'm not able to see the. Yes, group 1. Yes. So it's not moving. So here I wind up my session. I hope at least few points you have extracted well from this class. So it is not the class which is going to fetch you the complete information. The idea of the classes is these kind of classes is to make you orient to the subject and make you feel the competition and make you work more, make you study more, make you revise more. Revise, revise, revise. You repeat or any act, you do the hernia surgery 100 times, you become master of hernia surgery. Like that, if you read the same topics, facts, and you have a lot of facts are there in oncology. Even in chapter 28 of Sabiston, and if possible, you can read chapter 1 of Casciato, recent edition, Casciato. Okay, oncology, clinical textbook of oncology, clinical book of oncology. Chapter 1, that is more than sufficient. Please read it today or tomorrow, otherwise you will forget. And once again, I thank Dr. Vipin Sharma and DNX Academy for providing me this chance. And I thank you all for the patient listening. And I wish we would see you again in future DNX Academy classes. Thank you. Vanakkam.